Hey, good afternoon and welcome to everyone. This is the Health Research Board Trial Methodology Research Network webinar series. This is the second webinar of 2019 and we are delighted to welcome Dr. Ronan Fitzpatrick to today's webinar. The title for today's presentation is Innovative Sample Size Methods for Adaptive Clinical Trials. So Ronan is the head of statistics at Statsols, the provider and developer of nQuery sample size software. Leading through statistical innovation over 20 years, nQuery is one of the world's most trusted sample size and power analysis software packages. Used by biostatisticians at all levels of expertise, and with over 50,000 users in over 100 countries, nQuery helps its users quickly and confidently calculate the appropriate sample size for their study. In 2017, 90% of organizations with FDA-approved clinical trials used nQuery for their sample size calculation. So since 2013, Ronan has led the development of nQuery. During this period, Ronan has driven many technical and statistical advances in nQuery to respond to the statistical methods and trends of sample size determination. Recent innovation in nQuery have included tools for planning sophisticated adaptive clinical trial and sample size methods using Bayesian statistics. In addition to this, Ronan has given lectures and workshops on topics of sample size determination for many institutions, including UCC and the Food and Drug Administration. So before I hand you over to Ronan, I'd just like to turn your attention to the Q&A section at the bottom of the screen. So if you just hover over the bottom of the screen, the bar will come up there and you can see the Q&A box. Just to click on there and you could input your question and we'll put them all to Ronan at the end of the presentation. So without further ado, I'd like to hand over to you, Ronan, to start the webinar.
Thank you so much, and it's a great honor to have been given this invitation to give this presentation on innovative sample size methods for adaptive clinical trials. So the hope is in today's demonstration, today's webinar, to kind of give an introduction to adaptive design, in particular in the context of clinical trials, but hopefully keeping some of it relevant to preclinical research as well, and then hopefully also just give a demonstration of what kind of that practically looks like when you're planning a study using nQuery, but hopefully most of the lessons would be applicable regardless of what software solution you were using for the design process. So as mentioned here, like this is basically just reiterating what was there before. And like basically the last major thing we did was a computer technology workshop at JSM. So once again, just really thankful to have you here. And obviously nQuery was mentioned, the statistic here. So in terms of what we'll be covering today in today's webinar, we're basically looking at first a kind of overview of adaptive design and sample size re-estimation. So as well as like there's adaptive design is a very wide area, which is a lot of interest in clinical trials right now, both and preclinical research. And of course, what we're trying to do here is establish, well, what is adaptive design? How do we ensure that adaptive design is done in such a way as not to lead to biases due to unblinding and other situations, and also in terms of statistical criteria such as ensuring that the type 1 error rate is not inflated. And then we'll be looking at basically two types of sample size re-estimation, blinded and unblinded effectively, and then there'll be discussion inclusions at the end, including the Q&A. So just in terms, if you want an overview of what examples we'll be working through practically using nQuery, we'll be looking at a two-means blinded sample size re-estimation, a two-proportions blinded sample size re-estimation, a two-means group sequential design, which I'll go over as well, and two-means unblinded sample size re-estimation as well. So in terms of the first section, adaptive design and sample size determination. So I suppose for anyone who's not familiar with what adaptive design are or what adaptive trials are, an adaptive trial is effectively any trial where a change or decision is made to the trial while it is still ongoing in a formal fashion, I should say. Sometimes you can make ad hoc changes in correspondence with the relevant authorities and the relevant data monitoring committee, but this is kind of where you're planning ahead of time to make a change based on some criterion. So basically, you can think about this as a situation where if you can think of situations where you think, well, that's not what you originally were planning for, but that would be interesting to keep to change to ensure that we could find what we think is happening based on the current information. That's basically what we're looking at here. And there's a huge variety of different adaptations that you do. And I'll go over some of these in more detail in a second, but obvious targets for adapting while based on interim analysis, based on data as it becomes available, is something like increasing or potentially decreasing the sample size. increasing the sample size in particular subgroups, ones that have particular interest in the study, changing your hypothesis, changing which arms are or aren't included in the study, etc. And the hope here, I suppose, is that adaptive trials, as opposed to what we consider to be a traditional fixed-term trial, which is one in which we basically plan our study, we collect all of our data, and then we do a statistical analysis at the end. An adaptive trial is hoping to give you more control as a trialist, as a sponsor, to improve that trial based on all the available information, including the information which has been accrued so far. So I suppose from like a philosophical point of view, like if you knew what was going to happen in your trial beforehand, or if you knew how effective, say, a proposed treatment was, both a priori, then you could, create the perfect trial to get the perfect amount of evidence to move on to the next stage from a regulatory point of view, or from a sponsor point of view, or from getting a paper published point of view. And I suppose an adaptive trial is trying to move you closer to that platonic ideal of what that trial would look like, by basically using the interim information as a proxy for the best guesses that we have for a planning perspective. Because of course, in a traditional planning situation, including sample size determination in general, we're basically having to guess what we think is going to happen and build a trial around that kind of situation that we hope to occur effectively. So that's it's kind of what we're trying to get at here. And the hope then is that adaptive trials can decrease the costs, because basically you have more flexibility rather than having to kind of set everything around, well, we're going to run this trial and have no opportunity to change it while it's still ongoing, or minimal opportunity to do so. If we have the opportunity to change things, we can explore more options, we can include more groups, we can do things like that, and then basically have a more efficient or broader trial and getting more things done at once, and hopefully also lead to better inferences, because we won't end up with a study that's underpowered, for example, because the sample size was insufficient. So this is just a very brief summary of what are considered to be the major advantages and disadvantages of adaptive trial versus a more traditional trial, or basically versus a fixed-term trial. That is the trial where we basically set and forget. We do our plan for a study, we get our data, we analyze our data. There's nothing happening in that kind of middle point in terms of decision making. The advantages are obvious in terms of earlier decisions. So even the simplest type of adaptive trial is one where you could just stop the trial early if there's strong evidence for or against you actually getting a significant p-value, for example, at the endpoint. The reduced potential costs, both from being able to make earlier decisions, but also potentially being able to do more with any given trial, basically get more bang for your buck for a given trial by, say, including additional arms that wouldn't be viable if they had all be completed to fruition. Otherwise, higher potential success, it's very much related to those previous things in terms of basically having this opportunity to kind of look at more things and evaluate them greater. And of course, greater control is kind of what trialists are most interested in, because I suppose, to a certain extent, when you're setting a design and you're kind of not empowered to make changes to it, you're putting a lot of faith in those initial guesses, estimates, ideas about what might or might not happen in the trial in terms of when something actually happens. Now, assuming, of course, that an effect of interest actually exists. And then seamless designs, this is probably more of a technical issue for people who are involved in clinical trials for drug approval. But there's a big thing there where, obviously clinical trials go through several stages: phase 1, phase 2, phase 2A, phase 2B, phase 3, phase 4 / post-marketing surveillance. And the idea with these seamless designs is that you can integrate those into single data. So basically data from, say, phase 2B can be used in the subsequent phase 3 trial, rather than having to get a whole new set of subjects, a whole new set of clinical centers, etc., and hopefully increase the efficiency that way. And obviously you get a more longer comparison for that subset of people from that phase 2 trial, so you get even more information from them than you would otherwise. I suppose from a disadvantages point of view, obviously an adaptive trial will generally increase the complexity versus a fixed term trial, both in terms of the kind of statistical test statistics might have looked like, which is kind of more 0.3, but more importantly, probably in terms of having to think more deeply about type 1 error, type 1 situation. So for example, take the most simple type of adaptive trial called a group sequential design. This is where you have the opportunity to stop your trial early, 1, 2, 3, possibly more times, at what we call interim analyses, right? So let's say 50% of the people have been accrued. You now have the opportunity to stop the trial early if there's very strong evidence to do so. Like, let's say, you know, if you have a very strong effect size so far. Well, the problem with that is that, you know, obviously you're dealing with, if you have two chances to get a successful result for your trial, then under a frequentist paradigm at least, then you're kind of cheating. You're getting multiple chances to be successful, and that means that your overall chances of being correct or wrong based on statistical criteria on type 1 error, for example, is more likely to be inflated because you have these multiple chances to be right effectively versus the kind of fixed term trials which you're kind of comparing to implicitly. So that's kind of one of the issues. I think from especially when you're getting into clinical trials, logistical issues are a massive thing. In particular, blinding, if you're going to do an unblinded or comparative adaptive trial, that's one where you're actually going to look at the labels, like who is in the treatment group, who's in the control group, who's in whichever group is in the trial, then the blind is obviously a huge issue from a regulatory point of view. And obviously you have to create Chinese walls and other types of controls to ensure that there's no leakage of information to participants who aren't supposed to have access to that information. For example, this basically, like I'd say in adaptive trials, especially comparative ones or unblinded ones, there's a much higher emphasis on the importance of the Independent Data Monitoring Committee, which is an external body who will be basically associated with any phase 3 trial, for example, but are even more important in the case of adaptive trials because often they won't just be monitoring the data, they will be making decisions based on what they're monitoring thus far. Point 3, as I mentioned, like modified test statistics. So oftentimes in an adaptive trial, you may not be able to use the same type of test statistics as you would use for the fixed term trial. And therefore, there might be questions about how comparable the results are from an adaptive trial to, say, pre-existing fixed term trials, which have evaluated the same treatment or outcome. And obviously, because of all these kind of issues, there are greater resource expectations in terms of statistical expertise, and obviously just people involved in the wider logistical issues. You need basically people who've done it before, preferably, to kind of make sure that these issues 1 to 3 don't occur. And so the 5th point is worth pointing out, and I'll actually go into that in detail because there's some recent developments that is just like regulatory approval. This is becoming less of a disadvantage and maybe is becoming an advantage over time, but right now, I would say, an adaptive trial will require you to do more regulatory work, more interactions with a regulator or with other bodies to ensure that you have, done your homework and that the design doesn't have obvious flaws versus, to say, the more simpler fixed-term trial. So to give you an idea of what type of adaptive trial designs are out here, these are kind of 7, lists of 7, which is taken from the guidance I'll be talking about in a moment from the FDA. Basically, these are kind of 7 of the most common types of adaptive trial, but let me be clear, this isn't a full survey of them. There's a lot of more complex ones, particularly in earlier stage designs, like continual reassessment method designs, Bayesian designs, more complex ones. But these are probably the standard ones basically out there. So firstly, it's group sequential design. We'll be talking about that later, but basically you have the opportunity to stop the trial early for efficacy or futility, or basically very strong evidence at an interim point for efficacy or futility. Sample size re-estimation, which we'll be talking about in detail during the rest of this webinar, so I won't go over here, but basically you're able to increase the sample size adaptively, and adaptive just here stands for in response to interim data, in response to data that's occurred while, like we've accrued while the study is still ongoing. So let's say after 50% of the people have been recruited and have data available. Enrichment designs, these are quite common. There's a lot of interest in them right now. And these are basically where you can increase the sample size selectively in certain subgroups based on, say, them having a stronger response rate than some of the other ones. So let's say you're evaluating a very wide study and there's certain subgroups where you're getting a better result. Maybe you change the parameters of the trial so that you can focus on those subgroups going forward. Selection designs, this is where, let's say you have multiple study arms, like 5 or 6, and then you want to have at some point choose to pick out the 2 or 3 best arms. So for example, if you were evaluating different doses of a particular treatment, of a particular drug, then of course in that situation you might start off with multiple doses, and in these type of designs can allow you to drop the doses which are performing poorly versus the other doses that are available, and kind of keep the ones that are worthwhile and continue on the trial just with those ones going forward. Allocation designs, where you can allocate new patients adaptively. So let's say you want to ensure there's covariate balance, then you could adaptively ensure that people are allocated to the treatment and control group so that there's, roughly the same kind of covariate balance in each of those groups. Although the value of covariate balance is another debate entirely, but it is something that is out there. And endpoint designs, which is basically where you could change the statistical endpoint adaptively. For example, switching from an inequality or superiority hypothesis to a non-inferiority hypothesis. For example, I think there was people on Twitter talking about design where they found non-significance, very, very, very like 0.06 p-value, and basically the conclusion that they made or may have been forced to make by JAMA was that there was no effect. And some people were saying, well, if you've been allowed to use a non-inferiority hypothesis, that was only one of very many ideas out there. But if it had been a non-inferiority hypothesis, it would have been fine, and that would have been interesting in and of itself. And if you had that option to switch, then maybe that could have fixed things. And then obviously there's just multiple or complex type designs where you could be doing multiple of these types of adaptations in the same trial, or including other types of adaptations which we don't have time to go over here. So just in terms of the regulatory background, and I apologize, like mostly we sell to people who are based in the US, or who the FDA, the Food and Drug Administration in America, is considered to be the primary regulator for biologics and drugs effectively. So there's a bit of an American slant, I would say, from a regulatory point of view, but take that what it is. They are kind of the standard bearer or the most important regulator, even for manufacturers of drugs and other treatments in Europe. I suppose like one of the first things that happened in terms of regulatory guidance on adaptive trials was the FDA published their, CBER, that's the biologic and drug groups in the FDA, published their guidance in 2010. There's actually other guidance from other groups in the FDA on adaptive design. So in terms of medical technology, but that's kind of a different area. We mostly focus on drugs and biologics. And basically, I suppose the big thing that came from that, or maybe I suppose a big controversy within the adaptive trial community, is that the FDA effectively created a delineation between what they called well understood and less well understood designs. So there was basically a situation where people believed that if your design was in the less well understood situation, that your chances of getting approval from the FDA were relatively limited, and therefore the innovation that could have occurred in adaptive trials, particularly for confirmatory phase 3 trials, which is where obviously the vast majority of the expenditure and expense comes in clinical trials, that was limiting innovation in that area. And the EMA had a similar paper in 2007, though it wasn't quite as prescriptive in this. Despite this, there's been an increase in interest in encouraging adaptive design, I suppose for many of the reasons I've just discussed previously, in terms of their potential advantages. And there's actually been legislative attempts or regulatory attempts to try and increase the openness of agencies, the EMA and the FDA, to these kind of adaptive designs and other types of innovations in clinical trials, just due to the general understanding that the costs of clinical trials are escalating so large that many are arguing that that's hindering drug development and other developments in other areas. So in the US, you had the Innovative Cures Act. In the EU, you had this Adaptive Pathways Program. And sorry, I made a mistake here. This is from an old slide. But basically, the FDA published in 2018 its new draft guidance on this, and it was much different. I suppose that's worth going in detail here. So just in point, this is in late 2018 in October, and the FDA published its updated draft guidance. So remember, this is draft guidance. This isn't the final guidance, but the draft guidance is usually a good indicator of what we can expect to come down the road. I believe they were taking comments until the 8th of November last year. But obviously there was a legislative requirement under that Innovative Drugs Act to update this guidance. And just to mention, there are other individual guidances for certain types of design out there as well, like basket designs. But basically, I suppose the major takeaway now is that versus the 2010 draft, it was far less categorical. This less well understood delineation had been gotten rid of. And what they'd really chosen to replace the emphasis on was early collaboration with the regulatory agency, with the FDA, effectively. And basically they're saying, look, we realize that adaptive design is something that's out there, and as mentioned here, has the potential to improve study power and reduce the sample size and total cost for investigating drugs, including targeted medicines. But they said, look, if you're going to do adaptive trial, come to us early, and we want you to focus on design issues. We want to ensure that you think about your design, and obviously just think of the issues such as type 1 error, like that your alpha level. And I think for them, there's certain requirements that they have that aren't necessarily like required from a statistical point of view. One of them is pre-specification, which is to say that if you're going to have an adaptive design, you need to pre-specify what those adaptions are going to be and the criteria under which you are going to do those adaptions. And I think that's important because from a statistical point of view, if you go into the adaptive design literature, what you'll find is that in many cases, you don't actually have to do that. You can get away, you can basically make any adaptation that you want, and there are very statistically rigorous ways of making those changes. But I suppose from a regulatory point of view, the issue isn't the statistical stuff, it's more probably the ensuring openness point of view. It's ensuring that there's basically the lessening the probability of malfeasance, cheating, however you want to phrase it, in terms of being able to modify your trial in such a way as to make it more likely to succeed when it shouldn't, basically. Basically peeking at the data, changing things in such a way as to increase your chance of succeeding, despite the fact that without actually having had an effect worth doing something for. Blinding is also another situation, and I would mention, like if you're involved in this area, one of the big things that's coming, it's not a big thing, but it's just, it's mentioned that comparative versus non-comparative is probably the terminology that's preferred for precision point of view at the moment. And that's primarily just because we have stuff like open label trials where you can actually see the labels. So those are technically unblinded, but really comparative versus non-comparative. Comparative means you're going to be looking at the effect size effectively. Non-comparative means you're looking at some like nuisance parameter, like the variance. So I suppose from that point of view, blind means non-comparative, unblinded equals comparative. That's kind of terminology you might see if you're involved in the area. And just to mention that simulation is an expectation to show that all of the issues that we've talked about here in terms of your design issues, pre-specification, etc., simulation's a key part of doing that. And as I mentioned, I went into depth on certain adaptive designs, which I outlined previously, but they do have views on Bayesian and more complex designs. But I expect that to get fleshed out even further based on the responses from the comments window that closed in November. So in terms of what we're going to cover for the rest of this webinar, we're going to focus on sample size re-estimation as just kind of a subset of the wider adaptive trial thing, and hopefully illustrate some of the issues that come around here. I suppose, what is a sample size re-estimation adaptive trial? That's one where basically we're going to increase the sample size if it's required based on the interim evidence gathered so far. And it's an obvious adaptation target because much in the same way that, when we're designing a study, sample size is an obvious thing to increase or decrease to allow us to have a higher chance of, succeeding or getting a significant p-value or increasing our power or however you want to phrase it. It makes sense that, well, if we're kind of treating the sample size as the flexible component at the planning stage, then it makes sense that if we want to change something while the trial is ongoing, sample size would seem an obvious target for that. And just to note that, generally these things are better, like adaptive trials in general, and I would say sample size re-estimation in particular. It's easier to do adaptions if you have a knowable / short follow-up. That is to say that the time between when someone enters the trial and when we get information on them, let's say, your increase in blood pressure, or something like that, is relatively short, because what you don't want to have to do is be making changes, making decisions about the trial while it's still ongoing, and then have like a whole cohort of people who are like halfway through their follow-up period, and then you're not really sure what to do with them, right? And that's kind of the situation there. And just to mention that, you could adaptively choose to lower your sample size, but if we're talking about regulator point of view or sponsor point of view, usually the expectation is that whatever your initial sample size estimate is that you plan for your study, that's kind of the lower bound, just because I think that's just kind of what people expect. But there's nothing theoretically stopping you from decreasing the sample size as well. And as mentioned, there are two primary types of sample size re-estimation, unblinded and blinded, and they really differ on two aspects. One is that there obviously is the obvious one. Are they blinded? Are they unblinded or blinded? That is to say, is the effect size known at the interim analysis? Are we going to know who was in the control group, who was in the treatment group? Do we know the labels each person has? But the other thing is that they end up targeting different aspects of the uncertainty that you might have had at the initial sample size determination stage. SSD just stands for sample size determination here, just for reference. So we'll start with the relatively simple one of blinded sample size re-estimation. And when it comes to blinded sample size re-estimation or non-comparative sample size re-estimation, what we're focusing on here is we're not going to be looking at the labels, of course, that kind of comes with the definition. We're not going to know who's in the control group or the treatment group. Based on this data, we're just going to get a lump of data without knowing who's in each group. And what we're going to focus on is getting a better estimate of what we call usually nuisance parameters. That is to say, parameters that don't tell us how big the effect is, but which tell us some other information that was important to kind of get a reasonable idea of the sample size. And usually that's some kind of measure of the uncertainty, the variance, basically how much do we expect things to vary on a person by subject basis or a group basis. And I would say if you're going to do a blinded adaptive trial in general, and including blinded sample size re-estimation, there's a much lower barrier from a logistical, a regulatory, and a statistical point of view to do a blinded adaptive trial. If you're able to get an adaptation without having to unblind your data, that will reduce all of these costs significantly and reduce the chance of things going wrong significantly as well. Of course, assuming that you can maintain the blind, that obviously increases as long as you need to obviously have logistical plans in place to ensure that blinding is maintained in your trial. And for this reason, blinded sample size re-estimation was described as a well understood and attractive choice for adaptive design in the previously referenced FDA guidance. There are multiple methods out there for doing blinded sample size re-estimation, but the one I'm going to focus on here is the internal pilot approach, and that's primarily because it has a number of very positive attributes. One is that it's very simple and it's very easy to conceptualize. And two is basically you can kind of do this in a way that doesn't require you to really change your current workflow very much. So basically what we're going to do with an internal pilot approach is to get an updated parameter for a nuisance parameter. So an updated value for our nuisance parameter estimate, say the variance for the standard deviation, from what we call an internal pilot. So an internal pilot is really, like, let's say take the first 10% of the people who come into your study, call them an internal pilot, which is obviously just it's very similar to the idea of doing a pilot study in general. We're going to take the first 10% of people, call that the internal pilot, calculate an updated value of our nuisance parameter, let's say the variance or the standard deviation, and then re-estimate the sample size based on that. And then what we can do is if we've done that in a blinded fashion, if we've maintained it the blind, you can actually then use that data from the internal pilot and all the subsequent data, whether the sample size had increased or not, combine them into a single data set at the end and simply use the exact same types of statistical methodologies that we would have used if it was a fixed term trial. That's to say your t-test, your chi-squared test, your linear regression, etc. You do your internal pilot, as long as you maintain the blind, you get a better sample size estimate than you would have got otherwise. And then basically at the end point, it's as if the internal pilot never occurred, right? Obviously you'd have to have done, justified this regulator, ensure that your blind had been maintained. But from a data analysis point of view, nothing has really changed versus having done no blinded sample size re-estimation. I suppose for that reason, it's a very promising, a very positive choice, because obviously that just means there's less issues with having to, think about using different test statistics and stuff like that. And I suppose there are other methods out there, but they generally do require the usage of alternative statistics. They basically often use what's known as p-value combination, which kind of comes from Fisher's rules for combining p-values. And they're they have a much more strict error control than internal pilot, where it's more, you can show that the error inflation, like type 1 error, your alpha, is very minimal. But the p-value combination has strict control. But for the logistical reasons, for all these kind of nice positive aspects you get, the internal pilot might be just preferred for practical reasoning. So in terms of examples of doing blinded sample size re-estimation, I'm going to be doing 2 practical examples in nQuery here in a moment. We're going to look at 2, like basically we're going to do a 2 means blinded sample size re-estimation. So imagine a 2 sample t-test, for example. And in this one, what we're going to do is we're going to target the overall variance or the standard deviation. Of course, the standard deviation is just the square root of the variance for the t-test case. And that's the within group standard deviation. So the, the standard deviation in each of the groups, which we assume to be equal. And then there's multiple methods for, well, what's the best way of estimating the within group standard deviations given the blinded data? Because obviously there is some effect on the variance based on the amount of difference that is between the 2 groups. But generally, what I'll be able to argue we can argue fairly successfully is that the effect of the 2 groups being slightly different in terms of their means has minimal effect usually on the variance per group, basically. And then we're looking at 2 proportions where we're going to target the overall success rate. So, because obviously we aren't going to know who's going to be in group 1 and group 2. We're just going to look at how many people succeeded in general. What was the total proportion for everyone, regardless of what group they're in. And then if we can assume the effect size is true, we can actually get an update of the sample size. So assume that, say, the proportion difference is still 0.1. Given the overall success rate, we can kind of reparameterize what our updated expectations are for the group 1 and group 2 proportions. These are available for all types of hypotheses: inequality, non-inferiority, equivalence, etc. And so the one note I would make is that if you're doing inequality testing or superiority testing, usually you're pretty safe to use internal pilot with minimal kind of justification. For equivalence, non-inferiority, the error inflation can be more significant, basically, than in other cases, I would say. So that's kind of going through the hopefully a background and stuff. And I'll be going through the background of unblinding in general. But I think what we're just going to do now is just go through a brief demonstration of what blinded sample size re-estimation looks practically using nQuery. But as mentioned here, many of these things could be done in other software if you're interested. But obviously nQuery kind of makes it a lot easier because it's a GUI-driven software. So in terms of this is just a standard 2 sample t-test sample size justification from the New England Journal of Medicine, comparing cyclophosphamide versus placebo in scleroderma lung disease. Basically, in this case, the mean difference was expected to be -9, the standard deviation was 16, and they had a target power of 90%, and they needed 160 patients. But note that that's after accounting for a 15% dropout rate. And, of course, I'll do the very simple sample size calculus, the adjustment for dropout to get to 160 during this example. So this is what nQuery looks like. If you have any issues, if you can't see stuff or if I'm going too fast, feel free to do Q&A to give feedback on that as well. But basically, in nQuery, each row, basically all of the sample size calculations are occurring in this top left window. Each row corresponds to an assumption that's required for the calculation to occur. So for example, let's say our test significance level. So that's obviously just the the alpha below which you would find for significance. So you need a p-value less than this value to find for significance, basically. And then there's lots of other things like what is the group 1, group 2 mean? What's the standard deviation? And then we have some other stuff down here related to the internal pilot. Each column is an individual calculation. I'm not going to go through the basics of nQuery today, but if you are interested after today in knowing more about how to use nQuery, there's lots of videos available on the nQuery website, and I'll be happy to share any links for that. Basically, what we're going to do in column 1 here is just fill in first those assumptions that were used for the initial sample size calculation. So they had a 0.05 significance level. We'll just set this to 0 and 9. This is a proxy. These are actual optional elements, but for now, the important thing that comes out is that the difference in means is equal to -9. This is just one of those cases where you don't actually need all the information. You can kind of use some of the basic information. And just so you know, this is the latest version of nQuery, like nQuery Advanced. But this will work fairly similarly. Basically, the way that this works isn't very different from previous versions of nQuery, hopefully. The common standard deviation, so it's called initial here because this is our initial guess estimate for what we think the standard deviation was going to be, in this case 16, giving an effect size of 0.563. And, for 90% power, they got 136. Yeah. That's 136. And if we take 136 and divide that by 0.85, we should get back 160, or approximately 160. So that's how we get there. So just a small note, that one of these first things you'll learn in sample size camp, for lack of a better term, or if you ever do a course in sample size, is that whenever you do an initial sample size calculation and you want to adjust for dropout, the way to get to your adjusted sample size, that is, increase to account for the possibility of dropout, is to take your initial sample size, in this case 136. This is how many we actually expect to finish the trial. Was what we obviously recalculate in the sample size software. You divide that by 1 minus the proportion dropout. So if there's 15% dropout, that's 1 minus 0.15, and that equals 0.85. So we took 136 and divided that by 0.85. So don't, when you're going from like the after dropout to the post, like to the after dropout to the, from the one that's what you actually expect to get to the end, and you want to get to how many people we need to have that many people, assuming a dropout rate, you need to divide by 1 minus the proportion of people you expect to drop out. And obviously the proportion is just, 15% divided by 100. So just, your percentage divided by 100 gives you back the proportion. So you don't multiply by 1.15, for example, or 1.1 or whatever. So for 10% dropout, you would divide by 0.9, for 5%, 0.95, at 20%, 0.8, etc. Just a small note, it's kind of a common mistake. I just want to make something I'm encouraged to emphasize whenever it comes up. So let's say then we had a fairly small internal pilot in this case. So we this is what our initial sample size is going to be. We've estimated that we think we'll need 136 people in total, or 160 after accounting for the fact we think 15% of people are going to drop out. So there's going to be 136 people at the end of the study after dropout. And now we want to say, well, we think the standard deviation was going to be 16. Well, let's say we took the first 20 people and we said, okay, what we're going to do is we're going to take the data from those first 20 people. We're going to then calculate an updated estimate of what we think the standard deviation, the variance, was actually going to be. And in nQuery, you can enter that manually. You could just calculate the standard deviation, say, outside the software or wherever you want to do it. It's a fairly simple calculation most times. And you could just enter it here directly. But what we're going to use instead in nQuery is just a side table in nQuery. So a side table is just a small kind of helper function or helper table that appears below the main table when a relevant row is selected. And all this is really doing is giving it the opportunity to enter our 20 data points and then calculate an internal pilot standard deviation. That is our updated estimate for what we think the standard deviation is going to be, or what we actually think it actually is, I suppose, in the total population or what we expect in our study. And I've just prepared like a small data set here, which was actually simulated so I actually simulated this data based on the actual assumption of the standard deviation not being 16, but being 20 per group. So this is just a small simulated data set I've done previously. If you want to go, it's very trivial code, if you want to, we can certainly facilitate that. And so we enter our data set here, and then it automatically calculates the standard deviation based on this data, which has come from the first 20 people that I simulated. So this is obviously, artificial data, but it says that the internal pilot standard deviation is actually, closer to 20 than 16. So obviously we simulated like that, and that's why unsurprising. And note that there's multiple options here for what you want the variance to be. I don't have time to really go into them in detail, but basically, surprisingly, based on the research done by Kieser and Friday, who kind of are two of the kind of people most involved in this area, probably two of the people who are considered experts in the field, is that you don't one of the simplest ideas that you would have, which is to just take, well, I have this 20 people, just take the overall variance and assume that's the per group standard deviation or per group variance, actually works really well. It actually is pretty much, from their perspective, based on their research, the most efficient way of doing it. Like if you're trying to just balance having roughly the same amount of power that you did beforehand. So in that case, that's the default assumption, but there are other assumptions like a bias adjusted one, which kind of tries to account for what I talked about there, like that there's bias introduced by the fact that there's a mean difference. Basically there's a well-known approximation by Zucker, or the upper confidence limit if you were trying to be conservative. But in this case, like usually I would say the conservative is unwarranted. And so you can see here that the internal pilot standard deviation was set to, closer to 20. And obviously that variance is higher, that standard deviation is higher. And for that reason, the sample size, if we had done this initially, would have been higher as well. And so you can see that the re-estimated total sample size is 208 versus 136. So it's increased the amount. And just divide that by 2 for the sample size per group. So that's 104 people per group. And you can see here that it just gives 2 options here for the stage 2 sample size. That is to say, the sample size that will occur after the internal pilot of 188 and 180 in each group. That's just obviously the total sample size is just 20 plus 180, which kind of emphasizes that this sample size and this sample size at the endpoint are basically going to be combined and used as if they had, been in the same study all along, had basically treated no differently. And the restricted and unrestricted thing only really becomes relevant if the variance, the standard deviation that you find in your internal pilot is less than your initial estimates. Let's say this was actually 10, and you can see here that this does make a difference. So the re-estimated sample size, if we had a standard deviation of 10, is 54, which is less than 136. So it's like 27 per group, I believe. And for the restricted case, you actually, the lower bound is the initial total sample size. So if we initially said we're going to have 136 people, we have to have 136 people under the restricted design. And in the unrestricted design, you have the option to decrease your sample size from your initial sample size estimate and do so. As I said, if you're kind of doing some stuff on your own or preclinical research, you could probably decrease the sample size if you talk to the right people. But obviously, like mostly, I'd say nQuery, we generally mostly deal with people who are doing like phase 3 trials, confirmatory phase trials. And in that case, the regulator generally looks more askance at reducing the sample size versus increasing it. And so what I want to just do very briefly here is illustrate the same type of design, but for where we're comparing two proportions, say using a chi-square test. And basically, for practical purposes, this is going to look very familiar for this. Basically, once again, we have a first section, which is just setting up the inputs required for a 2-sample chi-squared sample size calculation. And then we have some additional parameters required to define our internal pilot approach. So in this, like I only showed it very briefly there, but it's in there, the slides where this example is from. Basically, there's a 60% success rate in the control group, 90% in the treatment group, and they estimated that for a 2-sided significance level, 80% power, they would need 32 people per group. So we have 5% significance level, 0.05 significance level, 60% or 0.6 success rate in the control group, 90% in the treatment group, odds ratio of 6, so quite a high odds ratio in this case, if I'm being honest. This is quite obviously they were quite optimistic about their proposed alternative. And for 80% power on a sample size ratio of 1, which just indicates obviously equal sample size per group, you get 63. Now, obviously they give 32 per group, which is obviously 64 in total, but they probably calculate on a per group basis. And you'll obviously, for rounding purposes, if you calculate on the basis that you calculate the sample size per group, it has to be an even number for total sample size. This is probably actually the exact sample size required. We'll set it at 63 for now, just for convenience sake. Let's say again we had an internal pilot sample size of 20, and we don't need a side table in this case because the input that we're using, like the nuisance parameter that we're using to do our blinded sample size re-estimation, is the overall success rate. And obviously that's relatively easy to know. We can either enter the proportion of people who had the event, or we can enter the number of successes that were had, and we'll automatically get the other parameter from that. And we could say here, well, look, like the midway point between 0.6 and 0.9 is 0.75. So that's probably our working assumption, say, for what the overall success rate would be based if these were actually exactly true. But let's say the actual number of successes was closer to 70% than 75%. And you can see here that that is equivalent to around 14 successes in total. It's actually exactly 14 successes in total. And you can see that if that was true, if we assume that the proportion difference, this value, or the odds, well, say the proportion difference is still exactly 0.3, we can recalculate the what we think proportion 1, proportion 2 based on the overall success rate are. And if that is true, then the overall sample size would increase to 71 in this case. And obviously that's not a substantial increase. I would say that the overall success rate, using an unblinded sample size re-estimation requires you to assume that this proportion difference is exactly the same as you pre-specified to be. So I think the assumptions for the proportions case are much stronger than for the means case, because I think the variance or the standard deviation can be considered to be more of a true nuisance parameter than the overall success rate. But hopefully that kind of quickly illustrates that for the proportion cases works fairly similar. And there's similar results available for other designs like survival, Poisson type models, negative binomial, etc. So just for the last 15 minutes or so we have here, I'm just going to go through group sequential design, conditional power, and unblinded sample size re-estimation, and do a very quick example of what that looks like, and hopefully draw some of the differences or similarities between them. So just to know what a group sequential design is, a group sequential design is one where we have what are called interim analyses, which are pre-specified. I should emphasize that. So basically, let's say between a third of the people are occurred, the next third, and the final third, and then we have tree analysis, for example. And these are happening while the trial is still ongoing. So like after a third of the data is accrued. And obviously, you know, what we're trying to do here is to assess, in a standard group sequential design, we're trying to assess whether based on the data so far, and remember we're looking at, this is an unblinded type, a comparative type of adaptive trial. So we're actually going to look at who was in the treatment group, who was in the control group, get an effect size, so get the mean difference and the variance and calculate like a Z statistic or a T statistic, etc. And we're going to say, well, based on this interim evidence, is the chance that we're going to get a successful p-value at the final analysis based on the evidence so far so high that we can basically say it's more or less, you know, the chance is so high that we're happy to basically stop the trial early and say it was going to succeed in the future. And if we don't have enough evidence to do that, that the effect is either really strong or very weak, so you can stop for benefit, which is to say that the interim evidence is very strong for your treatment, or we can stop for futility, which is where the interim evidence is very weak and saying that it's unlikely that you will get a successful result, like a significant p-value, for lack of a better term. Those are your two options, and you can have the option to do you can at the same time have the option to do both, or you could just have one of those available at each interim analysis. And obviously, if you don't fulfill either of those criteria, you just continue to the next look and then decide whether to make the decision. And at the final analysis, of course, we always by default are making a decision like that. Because if we have a significant p-value, we're really finding for benefit or efficacy. And if we don't, then we're finding for futility. And the way that we usually do this is what is known as an error spending approach, where we're spending our alpha error, our type 1 error, and beta, which is our type 2 error. So type 1, obviously, is just our probability of not rejecting null hypothesis when we shouldn't. And beta is obviously the other way around. And just like here, just a small statement from the FDA guidance outlining what some of the advantages of group sequential design are from that FDA guidance I referenced earlier. And, you know, as I mentioned, alpha spending is associated with efficacy bounds. Beta spending is associated with futility bounds. Here's just an example of a spending function. There's multiple options available, but I would say in general, people practically end up using conservative bounds. That is to say, ones which spend less error at earlier looks. So basically, the evidence that you would need at earlier looks needs to be very, very strong to be able to stop the trial early. They spend less at that. And that basically what the trade-off is kind of like the more error you spend early on, the less is left over for the final analysis. So, you know, if I spend a lot of error, if I have, you know, very liberal bounds, that is to say it's very easy to stop the trial early, that means that stopping the trial early at the final analysis will be much harder versus the equivalent fixed term trial, right? And I suppose the reason people are conservative is because the controversy probably comes in cases where, you know, you get like a p-value of 0.04, like let's say a p-value of 4.5%. You've used the group sequential design, which spent a lot of error earlier on, and now the p-value that's significant because you spent that error is the boundary is much lower. Let's say it's like 3%. And obviously people see, oh, I got a p-value of like 4.5%. Why isn't it significant? And it's because, well, you wanted to have lots more chances to stop the trial early, and you wanted to have a really high chance of stopping the trial early. We didn't stop the trial early. We got to the end. Our p-value would be significant if we just done a standard trial. But because we spent it earlier to maintain the overall error, we can't do that. I suppose reducing the probability of that happening, reducing that kind of distance between the p-value we start off with like 0.05, 5%, and the final one we have, people don't want it to be too large because we don't want to have those situations occur where there's more controversy. And obviously what we're just doing here, the more error you're spending basically at each interim analysis, the more conservative the final analysis will be, the harder it will be to get a successful p-value at the final analysis. So this is just an example of a group sequential design comparing anticholinergic therapy versus Botox for urinary incontinence. And once again, the details are in the slide here, both the statement from the actual paper and then just a tabular summary here. But in terms of how this looks practically in nQuery or other similar software, this is what it looks like. Basically, you could split a group sequential design when you're planning it into 2 steps. And that is basically your kind of fixed term trial inputs and your group sequential design inputs. So your fixed term inputs are basically the exact same things that we were using for that 2-sample, the blinded sample size re-estimation, right? So we still have an assumption that, the significance level is 5%. In this case, the group means are minus 2.3 and minus 1.5. These are change from baseline measures. And there's a whole other debate about whether change from baseline measures are good or not, but we'll ignore that for now. They had standard deviations of 2.1. So obviously these are all different inputs because it's a different design, a different study, but these look very familiar, right, from this previous example of what we were looking at here. And I would say in nQuery, it's generally recommended that before you do a calculation in the main table, which is to say you specify enough information that the calculation occurs, you should probably specify the group sequential design, which in this case was for a single interim analysis, which is a total number of looks of 2, and an O'Brien-Fleming design. So this is you have multiple ways of specifying what the spending function looks like, and multiple spending functions available. O'Brien-Fleming is a very common one. Hang-Shi-Dakani is another one. That's gamma family is quite common as well. Power family. Obviously, don't have detail to go into these in detail, but if you want more information, there is more available on our website. So that's enough to get us to the design initially put forward by these authors, and then we see we have 109 per group. And if you go back to the actual previous slide, it'll say 121 per group. But if you take 109 and divide that by the dropout rate, which in this case you can see was 10%. So we're dividing 109 by 0.9, so 1 minus 0.1, or 1 minus 10% divided by 100. We get 121 as per the paper. So just note that that's a small difference there. And just to set up what a futility design might look like. So this is what the group sequential design outputs look like. We have these, so basically this is look 1. This is our interim analysis here, happening after 50% of the time is gone. And basically it's saying if the Z statistic, which is to say, your mean difference divided by the standard error, hopefully that means something there, but your standardized Z statistic is basically above 3 or below 3, or more accurately 2.963, then you would basically stop the trial early for strong evidence. And in terms of if using like a standard t-test or z-test, basically we're saying if the significance level, if the p-value was less than 0.003, then we could stop the trial early at this point. And you can see here that because we used a conservative O'Brien-Fleming bound, the significance level required at our final analysis, where the Z statistic has to be, above or below minus 1.969 or 1.969 respectively, the p-value for a standard 2-sided t-test is 0.049. So obviously that's very, very close to the 0.05 or 5% level we initially were setting. But for more conservative designs, obviously things might get a bit more complicated. Just before getting into the unblinded sample size re-estimation, let's quickly show what a futility bound would look like. Let's just do the same thing, except we're going to set this to 0.025 at the one-sided level, just because that's the equivalent one-sided level to the two-sided level here, and add a futility bound, which is also, we'll say that's Hwang-Shi-Dakani and minus 1.5. I don't really have time to go through that, but that's basically halfway between a Pocock and like a conservative O'Brien-Fleming and a more liberal Pocock design. It's kind of like a halfway house. And for 80% power, we'll see the size requirement has gone up a little bit to 114. And in this case, basically what you'll see is that instead of having a lower bound, basically to just be clear here, upper bound in this case means effects in the same direction as the initially specified difference of means, which in this case was set to a positive value. Sorry, there's a mistake there. Well, let's say it's positive differences. So this means that if you have very positive differences, which are equivalent to a standardized statistic of 2.963, then you would stop early for efficacy. But if it was less than 0.508, then you would find for futility and stop the trial early, because there's very low evidence that you would actually get a successful p-value at the final analysis. And in this case, the nominal alpha is still pretty much very close to our initial significance level, right? That's the p-value you're really interested in at the interim and other analysis for the efficacy bound. For the futility bound, you probably just need to calculate the z-statistic. So we're running a little bit short in time at the moment. So what I'm going to do is just mention that conditional, like when we're going to talk about unblinded sample size re-estimation on the next slide, the usual criteria is conditional power, and the conditional power can basically be defined as how likely we think it is that we'll get a significant p-value given the evidence so far. So the power is like the power, its definition is like the probability of rejecting the null, like having significant p-value given what we think is going to happen. The conditional power is the same, but based on the interim like data, the interim evidence so far. And just to note, it can also be used for futility boundaries in a more ad hoc way as well. And basic conditional power is what's being used to decide whether to increase the sample size or not in an unblinded sample size re-estimation. And basically, like a group sequential design versus an unblinded sample size re-estimation, the typical way of thinking of it is that rather than a group sequential design just having the option to continue to the next look or the final look, or stopping early because the evidence is very strongly for or against you, you also now have the option to increase the sample size. And the idea here is that maybe you want to initially power for a more optimistic effect size, or perhaps what you expect to happen, but to still have the option to increase the sample size when you still have enough evidence that there is a effect size that would still be clinically relevant. So maybe the minimally clinically relevant effect size. You want to still have the option to increase the sample size and find that, but not have to put initial resources into finding that higher sample size for a more conservative effect size. And basically, you can basically increase the sample size for promising results. And this is why this type of design is often called the promising zone design. There's two main methods here, but I'm just going to focus on Chen, Demets, and Lan. And basically, the difference between these designs and two different types of options, the Chen, Demets, and Lan, and Kui-Hung Wang, are actually very similar to what we talked about in the blinded case, which is to say the Chen, Demets, and Lan, if you fulfill certain criteria, which is to say that the sample size re-estimation has to occur at the penultimate look. I just mentioned, I just opened this using this interim monitoring and sample size re-estimation button here on the side table in the group sequential design table. The Chen, Demets, and Lan, basically you can use pre-existing group sequential design, your p-values that you had previously, the z-statistics, all these, like, basically these, everything here, exactly the same as group sequential design. If you're doing it if you're doing sample size increases at the penultimate look, like the look just before the final look, and the result falls within a predefined boundary, which is usually either 50% to your target power, say 80%, or can actually be defined derived for a specific group sequential design using this option for the derived Chen, Demets, Lan lower bound. And the big difference in this design and the Kui-Hung Wang approach, which can be selected from this selection here in the sample size re-estimation method, is that in the Kui-Hung and Wang, those restrictions don't occur. You can increase the sample size at any conditional power. You can do it at any look. You basically have full flexibility, but it's based on a weighting approach similar to that p-value combination concept, which offers strict type 1 error control, but at the cost of, for example, the weightings have to be based on your initial sample size estimate. So what happens is that the people who occur after your sample size increase are basically theoretically being weighted less than people before it. And that's kind of where the controversy comes in, among other issues. So what we're going to just do here is a very quick sample size re-estimation. And there's a lot of options here, but primarily what we just want to focus on is that we can specify what the maximum total sample size could be. A typical value would be twice our initial sample size, which was 228 in this case. We can decide whether we want the sample size to increase until the conditional power equals our original target power, or we can increase it if a result is promising, just increase it immediately to the maximum sample size, that is this row here, 450, this row here. And the reason you might want to do that at the max sample size is if you want to, guarantee, like basically there's a chance if you increase the sample size by a specific amount, there is a chance someone could back calculate what the interim effect size is who shouldn't know that information. And that's a major no-no from a regulatory point of view. And that's why it's kind of considered important. And so what we do then, once we specify the rules for our sample size re-estimation, is we just enter what happened at look 1 and look 2. And in this case, let's just assume that the difference of means was actually 25% lower than expected. So remember, our initial estimate was that it was going to be 0.8 difference in the groups. Let's say it was 0.6, 25% less. And if the standard deviation in each group were 10% higher than expected. So they were initially 2.1 per group. Now they're 10% higher. That's 2.31. So that's just 2.1 multiplied by 0.1 is 0.21. Add that by 0.21, you get 2.31. And you'll see that we get a interim effect size, an interim test statistic, standardized statistic of 1.387. And that gives a conditional power of just below 50%, which fell between our lower bound that we derived here of 35.7 and our upper bound of 80%. And therefore, we increased the sample size till the conditional power equaled the target power. And in this case, that required increasing the sample size to 429. So we've increased from 228 in total to 429. So that's around 215 per group versus 114 initially. And we can see here that if we assume that effect size was still the same, so 0.6 to 2.31, we're just using side tables here, as we explained earlier, that's using a similar side table. We find that the significance level, this Z statistic of 2.69 was significant. And for this toy example, it's fairly trivial to show that if we had chosen not to increase the sample size, and obviously this is a toy example picked for the purposes of obviously this result's coming out, but if we'd done the exact same situation, deleted something there by accident. In this case, we'll say there was no sample size increase. Let's say the sample size increase didn't occur. We go back to our initial example, we enter the same values, and we would find that in this toy example, we would have had a non-significant test statistic because it's less than 1.969. And obviously in that case, the sample size estimation has saved the study, I suppose, in that case. Just the details of that sample are given here. So I think we've just run out of time there. Apologies for running a couple of minutes over. But basically, here's basically the conclusions that come with. Adaptive trials are becoming more common if you're involved in later stage clinical trials. They're already very, very common in preclinical research and earlier stage trial designs. And there's a lot of innovation happening there. Just like we at nQuery, we're mostly focusing on confirmatory trials because that's where sample size determination is kind of a standard design requirement. But we are moving more in that area as well. Sample size re-estimation is a very common type of adaptive trial. And then just here's some basic points about blinded sample size re-estimation and unblinded sample size re-estimation as well. And if you have any questions, the emails are in the slide, the references are here, and just here's some details on the last release. So just want to thank you all for attending, and apologies once again for running a couple of minutes over. So if you want to make some final comments there.
Great. Yeah, that was absolutely brilliant, Ronan, and so great to have those worked examples. Like, it really brought the presentation really to life. So thanks so much for that. I've got a couple of questions here that came in a bit earlier. And the first one for you is, are there any other methods of estimating the variance of a blinded sample size increase?
Yeah, so as I mentioned, there's 3 options given in nQuery, which are basically just using the standard deviation calculation, just your very simple one, the kind of bias adjusted one, which is very not too complex after that, and the upper confidence interval limit approach, which is kind of a conservative approach to it. Mm-hmm. But there are other methods out there. Gold and She have what's known as an EM approach, or an expectation maximization approach. But I think the recommendations that came from Fried and Kaiser, who are kind of the primary people we use, was that it wasn't really that good. But I have had some requests for it, so if it's something you've heard of, let us know, and we can consider adding it in a future release.
That's great. Another question we have here now is, what should the assumed effect be for a conditional power calculation?
Yeah, so I didn't have time to mention this as much, at the time, but basically there's two options. Either you can assume that the effect size, so just to mention, for conditional power calculation, you still need to assume what you think the true effect size was in addition to what the interim test statistic is. There's two main ways you can assume that to be. Either use your initial assumptions, or generally I would recommend using your interim estimates for the effect size. That is your mean difference and your standard deviation. Just, you can use those for conditional power calculations. So that's really the way they're thought about by the authors who use this unblinded sample size re-estimation approach.
Yeah, brilliant. And the last one was, where can you find the new FDA adaptive trial guidance?
Yeah, so as just in the final references slide here, it's the first reference just given here. And obviously that should, like, the slide should be sent on to you very, very soon.
Yeah, so we're going to have all this up on our website, up on the HRB-TMRN website. So you'll be able to see the whole presentation again and go through these slides then.
Yeah, I just got one more question from the Q&A tab, just asking, are there methods on unblinded sample size re-estimation focusing on nuisance parameters? Just this final one here.
Yeah.
And there are methods out there. We've kind of focused, we've kind of focused on the blinded one because, you know, the logistical and the cost, et cetera, is much less, but there are methods out there for that type of analysis as well. I think Potvin, for example, in the context of crossover trials, has talked about that. Like, yes, basically the short answer is yes. I can send you on references for types of designs that do that if you're interested. Munya.
Brilliant. Listen, thank you so much, Ronan. That was really, really good. And I'm really glad that you presented it today to us. So as I said to everyone there, the webinar will be available on the website. And just want to lastly say thank you again, Ronan, for joining us. And thank you for everyone else that tuned in from home. Keep an eye on the website. We will be announcing the webinar for March very shortly. So thanks again, Ronan.
Yeah, thanks so much. And hope you all have a good day.
Okay, thank you. Bye.